Hey guys, Mrs. Breha here. Another snow day upon us, so here we are, e-learning from home once again. Just an overview of the assignment that I have prepared for you guys today. Please complete all of these before 3 o'clock p.m. In order to be counted as present, and I'll also throw some free points at you for completing these activities. Number one, you should watch the Amoeba Sisters video that is posted on Schoology. Number two, which you're completing right now, is to take notes over the section we would have covered today in class. Number three is to review the online activity about mutations, which is our topic for tomorrow, and to ask 3 questions that you still have either about gene expression or mutations. And you're going to do this on the discussion board post right below this activity on Schoology. Now just keep in mind that everyone will be posting questions and you will be able to see other people's answers only after you ask your questions. So don't be alarmed if you don't see anyone else's answers. I want to see what you guys can come up with. The other challenge is to ask 3 different types of questions. Try to shy away from just the rote memorization type questions and try to dig a little bit deeper and really think about this information. These questions can be almost anything, and I've put a graphic with some sentence starters on there and also an example of 3 questions that I still have about this underneath this whole assignment. So please make sure you reach out if you have questions and make sure you get this done before 3 o'clock p.m. so I can have my attendance in before 4. All right, so here we go with our notes. So today's topic is about gene regulation. And think of genes as the instructions for making proteins, but we know that not every cell in your body needs every single protein in your genetic code all the time. So gene regulation is like the on and off switch for when and how much of your proteins actually get made. Keep in mind that every single cell in your body will regulate and express genes differently, and that's the primary way that we get differences between hair cells and skin cells and liver cells and stomach cells. So in order to save energy and resources, this is also done on a day-by-day or even minute-by-minute level for your body. This is the main way that most multicellular organisms help respond to their environment and help their bodies to grow and develop from a single fertilized egg all the way to a robust, mature adult organism. So gene expression, according to your book, is just this process by which a gene either produces its product to carry out its function, or it doesn't. So it can be an on switch, it can be an off switch. It really depends on the specific organism, the cell, and the individual protein. Also, it's really important to remember that there are significant differences in the way that gene expression occurs in prokaryotes like bacteria and eukaryotes like plants, animals, protists, and fungi. So in bacteria, we have studied this very broadly, talking about gene regulation and how most of the genes are located in sequence. So if it requires 3 different proteins to digest a certain chemical, then those 3 proteins will all be coded for by genes that are in sequence. That's 1, 2, 3 right in a row. This model is called the Operon model, and we can say that these genes all operate together as one operon. The best studied operon in bacteria is called the Lac Operon, which is created by bacteria that digest the protein lactose. And lactose is an inducible operon, which is a little bit fancier than what we need to know. But what you do need to know is that when there's no lactose present in the environment of this bacteria, then it will not express the gene for a lactate, which is the enzyme that digests lactose. So as soon as some lactose is found in the environment of this bacteria, it will bind to the repressor protein, and then that repressor protein can no longer bind to the DNA, and it works as kind of like a negative transcription factor that we saw last week. So as that repressor is removed, it then allows the RNA polymerase to bind to the DNA and express that gene, so then we can create lactase in order to digest that lactose. So this is actually quite a complicated process, but remember that bacteria, even though they are small, and relatively simple single-celled organisms, they're actually highly evolved and highly specialized for their functions. So that Operon model is only seen in bacteria. In eukaryotes like you and I, gene expression is actually even more complicated because we might need genes that are located far apart from one another on entirely different chromosomes to all get activated together. Okay, so in order for this to work, we instead used what are called transcription factors. And you guys saw these in your online activity last week. These are molecules that bind to DNA and allow it to be transcribed, which makes sense. And again, just like our Operon model, this can be turn on transcription, or we also have negative transcription factors that prevent transcription. But these transcription factors will bind to many different areas of DNA all at the same time and allow for the regulation to occur with some synergy, so they're all working together. So a typical eukaryotic gene has a regulatory sequence called a Tata box. And you can probably guess that that just means it is thymine, adenine over and over again. And this works as a promoter sequence. A promoter sequence is that location where the transcription factors will bind, and it also gives the RNA polymerase a little bit of time to begin that process of building what we call an RNA primer. So this is another way that eukaryotes, or scientists that go and look for genes to try to identify what it is that they do, are always looking for this TA hundreds of times over to identify when a gene is coming. There's that Tata box. We have the binding of that transcription factor, which will create the possibility of transcribing and eventually translating these genetic sequences into functional proteins. So lots of different ways that eukaryotes can turn on or turn off or create a larger amount of a certain protein whenever necessary. So, but the big idea here isn't really the how, but more the why. This is the main way that the cells within your body that all possess the same DNA can look different and have different functions. Gene regulation is what makes cell specialization possible, and you see lots of examples of highly specialized cells here. Muscle cells versus blood cells versus bone cells. They all contain different amounts of proteins that allow them to do their jobs, and the proteins that built them are going to be different, as well as the proteins that allow them to sustain those structures and functions. So the most important time in any eukaryote's life for this gene expression to occur is really during development. Development is where we go from being a single-celled fertilized egg into an embryo and then eventually into a full-grown human. The process that allows cells to change in structure and function is called differentiation. So on the left here, you can see a very early 4-cell embryo or 4-cell zygote. It still is at this stage. You can even see the shell, or we call that the zona, on the outside of the egg. We can watch human development happen in the lab because of fertility technology. We can see this happen and we understand it really, really well. So this egg was probably fertilized 24 to 48 hours ago by the time it's done 2-cell divisions. And then about 8 to 10 weeks later, it will look like this little creature that's made of millions of cells. And then 9 months later, it will look like this little creature, which contains trillions of cells. So that process is pretty amazing to understand how and why we go from one genetic code to this extremely diverse and differentiated type of organism. So another way that we use model organisms in biology is to study this differentiation process, because we know pretty well exactly how it works with humans, but we can't do experiments on humans. Obviously, that would be really unethical. So instead, we can study other organisms like fruit flies. We can do what's called the knocking out their genes, where we turn off some of these developmental genes to see what will happen. One type of gene that's really, really interesting to scientists are called homeotic genes, and these regulate when and where organs will develop. So you can see in our fruit fly chromosome where the different colored genes are and then where they cause different features to grow. So the green gene will cause the head to grow. The yellow gene will cause that thorax to grow, and the blue genes cause the abdomen to grow. And we can even see those same genes working in larger organisms and mammals like these mice. We also have Homeobox genes that work hand in hand, code for specific transcription factors. So again, we've used different types of technology to see what happens when these genes malfunction or if they don't work at all. And of course, we don't do those types of things on humans, but we do them on other model organisms and then apply that knowledge. And then finally, we have the Hox genes. The Hox genes are the genes that will determine the identity of each body segment. So the idea that the head is at one end and the tail is at the other. These genes are extremely important, and even if we're looking at just that 4-cell embryo, it doesn't take long before those cells have an identity and they have already been decided what they will become. Now, remember that your genes aren't the only thing that will affect your phenotype, the way that your body looks and functions. The conditions in an organism's environment will definitely affect their environment. We have some really interesting examples of this in the animal kingdom. Here we're looking at Himalayan rabbits, and in your book they talk more about it, that those raised in cold environments where the Himalayan rabbits evolved will actually have darker fur on these kind of cold parts of their body because that helps them attract more sunlight and to stay warmer, whereas those raised in captivity where they never get cold do not express these genes, and so they never get those telltale markings of a Himalayan rabbit. We've also seen this in tadpoles as they metamorphose into frogs. If they are in a stressful environment, so less water, less food, not as good at temperatures, they will actually undergo metamorphosis faster than those allowed to be in a more friendly environment. One up-and-coming type of research is called epigenetics, and this is really the study for humans specifically of how your environment and your behaviors can work on the way that your genes are expressed. So it doesn't actually cause mutations, which we'll talk about tomorrow, but it causes when and if your genes get expressed. These types of changes are actually reversible, but they can also be inherited. So it can be how what you do in your experiences during your life can affect how genes work in future generations. And this is really, really interesting because it does show how our genes do change their expression over the course of their life. If you are like this cute little caterpillar, and it will undergo a very extreme metamorphosis. So different genes are being used at different parts of the life cycle, and that is all impacted by the environment, about photoperiod, and if the if as the seasons are changing, so is the expression of that butterfly's DNA. All right, so some really cool stuff today. It's pretty deep stuff, so if you're interested in any of it, I would encourage you to read through it in your book. Just another review of what you are to do today. Watch that Amoeba Sisters video. You just did the notes, so you can check that one off your list. There's an online activity for you to go through. It's really quick, less than 5 minutes, to introduce this idea of mutations. Ask 3 questions. They can be really broad, they can be really specific, but challenge yourself to find some deeper questions. Okay? After you've completed that discussion board, you're all done for today. And if you have any questions, you can text or email me. Hopefully we'll see you tomorrow after this blizzard has passed. Bye, everybody.